everybody. I'm Mike DeHolis. I offered to lead a roundtable discussion, fairly informal discussion, about plant synthetic biology. And I thought maybe we'd start just with some introductions. Please just introduce yourself, and then we'll have an opportunity for more discussion as part of this session. So if we can just go around the room, please say your name and your institution.
My name is, I'm from Hong Kong University, and I'm from biochemistry department, year 1 PhD student.
Okay. I'm going to try and keep some notes here as we go and forward these to everyone. So your first name was?
Alan.
Okay. Was it Yang?
Yeah, that's right. Not Yang, Y-A-N.
Oh, sorry. And from?
Biochemistry department.
Of HKU. Great idea. Okay. Okay, thanks. Hello?
When we get from actually the first meeting.
That was a typo.
National Institute for Plant Genome Research. Ferman Federici from Cambridge.
Oh, sure. Recording. Let's see. Very recent.
Cambridge, is that right? Yeah. All right, Dr. Medford.
Oh, no.
Oh, Colorado State, sorry. Two S's?
Just the rest of them.
I always really think there's 2 S's in it and then 1 F.
All right, is that everyone? And
My name is Yuli Wang, Y-U-L-E.
okay.
I'm also come from HKUSD as a student.
Okay, in biochemistry. And as I said, I'm Mike DeHolis. I'm in a biological sciences department at the University of Alberta in Canada. All right, so if I can, I will email you the notes from this meeting if the organizers give me the email addresses. I don't think we received participant lists, did we? Okay. Sure. All right. Do you have a pen? All right. Welcome. We're just taking names of everyone involved in the discussion. If you'd care to give your name and your institution. Okay.
D-I-A-G.
Okay.
M-I-N. Yeah. K-A-N-G.
Okay.
It's China Agricultural University.
Okay. In that.
Yeah.
That's in.
China Agricultural University.
What city is that in?
Sorry?
Beijing. Okay. One of my best students came from China Agricultural University. Okay.
or botany? Can you tell me of science?
Botany?
Of course, this Institute of Botany. Can you tell me of science?
Oh, I can't. Okay. CAS.
Institute of Botany.
Okay. Where's that located? All right. Who else has joined us? Not on the list.
How's it going?
Good.
Tito Jankowski, TITO, JANKOWSKI.
And Mr. Nar. Is that right?
Yeah.
Okay.
I'm from Do-It-Yourself Biology,
And where's that based?
California, United States.
All right. Who else has joined us?
Me.
Yeah.
I'm Patrick.
Okay.
Boyle from Harvard Medical School.
Hoyle, was it?
Boyle, B-O-Y-L-E. Capsule for Life.
Okay.
Hi, I'm Jim Demara. Well, D-M-O-I.
Oh, K-I-M.
Yeah. D-E.
M-O-I. M-O-I-A.
No I. Tomorrow.
And I'm at the University of Edinburgh.
Yeah. Right. All right. Dave. Are you sure? Okay. All right. Yeah.
Alex Nielsen, A-L-E-C-N-I-E-L-S-E-N, University of Washington.
Okay.
Ingrid Swanson, INGRA, Swanson, University of Washington.
Okay. Both of those are in Seattle, right?
Hi, I'm Nat Dano from Third World Network.
Okay. Ness?
Dano. Third World Network.
And that's a NGO, or is it a? It's based out of.
Malaysia.
Okay.
Jennifer Ford.
I'm sorry, I didn't hear Corbis with a C. Corpus. Okay.
Tango Echo Bravo.
Tango. There we go. Thank you. I'm Mike, Delta Echo Yankee Hotel Oscar Lima Oscar Sierra. I have to spell it a lot. So, all right. Anyone else? Okay. All right. So I thought, as I said, this is a very informal discussion. But what we'd like to do, I think, as a community, is assemble some kind of a list of the resources and tools that are existing, those that are maybe being developed, and those that are still needed to really make plant synthetic biology possible or more effective. And then we can talk also in the context of what applications can be envisioned for synthetic biology. Those include, as you can imagine, basic research, and there's some nice posters on ways of using genes from plants and in plants in basic research to understand existing processes of developmental biology or physiology, and then, of course, applications to improving or increasing food production, quantity or quality, resistance to stress, and so on. Aside from food, of course, there's interest in production of bioproducts, and I've separated out energy from non-food and non-energy bioproducts, which includes things like fiber, pharmaceuticals, platform chemicals, and so on. And as well, we can imagine applications, and of course there are applications existing already for different types of environmental sensing and phytoremediation and so on. Have I missed any applications there? Artistry. Horticulture? Call it artistry. Do you want to clarify artistry at this point? Okay. All right. How about we say aesthetics? Okay. Okay, so as I said in the abstract book here, plant biotechnology actually has a fairly rich history, and you can argue that a lot of what people are calling synthetic biology, particularly in plants, has been going on in some form or another in plants as plant biotechnology for several years. And so in some ways, plant biotechnology is a little bit ahead of the curve of applications of synthetic biology in other organisms. But some of the real hardcore synthetic biologists say we have to make a clear distinction between synthetic biology and biotechnology. Yes, everyone's been doing biotechnology for 20, 30 years, but the difference is that synthetic biology goes beyond biotechnology in that there's the acceleration through the availability of new DNA synthesis techniques and so on, standardization of parts, which should lead to less redundancy and more efficiency, and the abstraction of systems in that you have modules that might perform a function within a plant or within a bacterium, and that module would be made up of several smaller components, such as an operon. You don't need to know what each of those components does. The principle of abstraction says, here's a black box. This black box, if you feed it glucose, puts out artemisinin or whatever. And that abstraction is familiar to computer scientists, engineers, and so on, not something that's really familiar to biology, where we train our biologists to understand every aspect of every component that we put into a plant, for example. Okay, so without debating the semantics of the difference between synthetic biology and plant biotechnology, but keeping those principles in mind, what tools exist and what are needed to make synthetic biology happen in plants? Ability to transform. Okay. Yes. Yeah, I was going to sort those out as you say them. Okay, ability to transform plants. Of course, we can transform plants. What do you mean specifically? How do you want to go beyond existing technologies?
Well, with hearing Daphne talk yesterday, it sounded like it's still not highly efficient. It'd be awesome if we could get super efficient.
Okay, you talk about increased efficiency.
Yeah, it sounds more like we can transfer plants than I can transfer plants. You can transfer plants.
Okay.
Using whatever.
Okay. So increasing, so the one of the things that goes along with synthetic biology is people talk about the garage synthetic biologist or so on. So you want to make plant transformation available to non-specialists. And probably as a side effect, it would be useful to increase ultimately the efficiency of transformation for a number of applications that even so-called experts or people who are already doing transformation do. We'd like to get those efficiencies up. It would allow us to do a lot more types of screening and so on. Oh yeah, thanks. Forgot about that.
Yeah, for the hobbyist, I think that's an awesome idea. Like plant packing, and you change.
So you're talking about letting the 18-year-old male and female go and transform plants?
Yes.
Glowing moths.
I have a problem with that. I have a big problem.
And you're mostly worried about the 15-year-old males, right?
The 15-year-old male, not 15-year-old.
I forgot altogether.
Okay. So there's a security issue. Let's talk about the security and ethics separately. No, it doesn't. It doesn't fit. I'll probably be okay. No one wants to stay here that long anyway. So when the power runs out, okay. Okay. Increased transformation efficiency. Let's talk about the safety and ethics separately. But increased transformation efficiency would allow a number of applications that aren't currently available. Also might remove some IP restrictions, which are a barrier to some applications of plant biotechnology. Anyone know of any existing initiatives towards this?
Sure, there's two issues. One is the actual transformation of something, the other is regeneration.
Okay, yeah.
So that's certainly the chromosome field, green chromosome field, seems like that's a clear step forward in the transformation side of it. There's still the issue of regeneration, what crops or what plants can be easily regenerated.
Right. So for non-specialists, generally when DNA is introduced into a plant, with some exceptions, it's introduced into a undifferentiated cell mass, which then has to be turned into a regenerated with shoots and roots turned into a plant that can then produce seeds and continue the normal life cycle. So there's two issues that Jim was talking about, introducing the DNA into the cell and then getting that cell to regenerate into a whole plant. Regeneration has been mostly neglected. I think in the early days, even before recombinant DNA, there was a lot of effort in plant tissue culture. I think that has fallen off a little bit.
Regeneration in case of legumes.
Okay. Yeah.
The biggest problem, and they are very recalcitrant, especially legumes. It's not possible to transform.
Yeah, it's very slow, right? If at all. Right, so regeneration certainly is something that does not lend itself well to these principles of generalization and abstraction and so on in synthetic biology or high throughput biology. They are, tissue culture is very much an art form, so it seems. Okay. Increase regeneration efficiency, including crops like legumes. Okay, we can probably separate the issue of transformation efficiency also from the issue of intellectual property restrictions. There is an organization based in Australia led by a man named Richard Jefferson, Cambia, whose objective, certainly independent of synthetic biology, is to develop tools for plant molecular biology and plant biotechnology that are free of intellectual property restrictions. And this organization has developed a set of vectors which are widely used now, plasmid vectors, which still have a lot of strings attached to them, but they're almost as good as you can get in terms of being free of restrictive intellectual property. And also, his organization came up with a method for transformation that avoided some of the intellectual property restrictions on agrobacterium-mediated transformation. Okay. So Cambia, even though it's not really considered part of synthetic biology, it is consistent with some of the ideals that are associated with synthetic biology, such as this idea of freedom of use and open source.
So under the underdevelopment colony, we've just got resources to put together some phytobricks, plant-specific biobricks, which I think is perhaps even more pressing than the microbial field because of the restrictions, the litigation in the plant field. We've got MTAs, which are really quite restrictive. And just to make those things available, drawn from the literature.
Okay. So what types of components?
Particularly promoters, other genetic regulatory sequences, but also proteins.
So resistance markers and
And our particular interest is in extracellular sequence.
okay.
Jim, you say that it's restrictive and very difficult to get to transport these, to say these photo bricks anywhere.
There's lots of stuff in the literature, and if you want to get hold of some of this, you have a quite restrictive negotiation. They're all, essentially most of the things are exchanged under FTAs, Materials Transfer Agreements, and so you have to negotiate with either the university or the company to get hold of these things, and you can't be forbidden from transferring them. So for like a Biobrick library, I guess we've got the example of like GMPs in the existing Biobrick library, and we have plant-specific floating proteins that we use, which are different, slightly different properties and targeted for various reasons. But if you have material that comes under an FTA, you're legally restricted from transferring it on, so you can't donate it to a public database. And you have plant field, they're very good stock centers for distribution of seed and DNAs, but if you can't use them, you're sort of stuck. So I think there's a lot of resources in the field which people don't have access to easily. And if you are setting up to you've got something you want to make, and you have to write to it in a half dozen places to get those components, you're going to be stuck there for, and certainly if you don't have the legal resources on your end to deal with the MTAs, so it's a real problem. From company to company, that's a serious negotiation.
Is that something that really needs to be sorted out before photo bricks can really become?
To be clear, he's not talking about the photo bricks that he'll be making, that they would be free of the MTAs. But
No, I'm talking about, because the MTAs are transfer of materials. It's not IP, it's separate, so you're still going to be constrained by IP issues if you're a company. But for academic use, in most countries you can probably get away with. And as long as you don't try and do anything commercially viable with it, you can still use these things. Probably not to watch yourself. But in terms of just getting access to materials, if, say for example, it's synthesized separately or constructed separately, but it's functionally equivalent to something which has been demonstrated to be active in some functional fashion, then you've got the material distributed.
Mike, a question for Biobricks. I know they have a big legal arm to try to deal with a lot of the legal things. And many of the components that Biobricks have were patented like 30 years ago, in which case they're largely off patent now, where a lot of these so-called, in Jim's terms, the phytobricks would still be under patent. So that really changes the equation. Could the legal resources of Biobricks be used to help phytobricks?
In the UK, can you not? I know in the UK you can pretty much use what you want from the patent database in an academic context, and you're free from litigation. But I don't know if that translates to the same thing in the same case in the US.
In the US, it's clear as mud. So technically, no one has gone against academic. So as we keep our academic hats on, but if you start to have a project, let's say a hot biofuels project that somebody is really interested in, you can be darn sure that Monsanto and Pioneer and Syngenta will come knocking on the door. And they will revoke any of the academic freedom. And when that has happened, at least in the case of the pharmaceutical compound, the universities have left the researchers out on the limb and backed away. They said, we're going to let them sue you and not us. So everybody knows. That's why it would be nice to have this up front with whatever legal resources Biobricks has to come to bear now so that we can not have to worry about that. So I think obviously one of the big areas is Biobricks.
But is the issue with the distribution of the bricks in the MTA, or the issue with the intellectual property attached to the sequences encoded?
They're two separate issues.
Right.
So you can't take material that's been transferred under an MTA and then just hand it out. You just can't do that.
Right. So the concern, what is the major concern with the phytobricks? Is it the distribution, which is associated with the MTA, or is it the IP associated with some conceivable phytobricks?
Well, step one is just getting access to the things. So
Get,
if no one can distribute anything, obviously that restricts
right.
the traffic of these important pieces.
Right. So if setting aside the patent and IP issues, then as far as the MTA goes and the distribution, you're saying that Cambridge isn't happy with you sending out DNA to whoever wants it without sending an MTA because that opens them up to liability and so forth, right?
No, we don't want to restrict
Right, right, yeah.
We're just going to put them in the Arabidopsis stocks.
Okay, so yeah, so distribution is not an issue for the phytobricks that you're going to create as phytobricks. Right.
But they'll still have IP restrictions.
Right.
They
If
belong to, the IP belongs
Right.
to someone. If you want to then use it to make money, then you've got to negotiate.
okay.
So is the agrobacterium and holistic technologies for transforming plant cells, I assume that's still under IP, and they're pretty, they guard that stuff highly?
The initial patents were probably filed, my guess is I think it was around 85, if I remember right, in which case they are probably starting to come off patent. However, the major players of the field, what's the proper terminology, somebody in the field will know this, where they patent one on top of another, and so they're kind of staggered, so even though the original ones are off patent, all the improvements are not.
This is not the right one, Santo.
This would be a Syngenta, just to be fair in terms of how we pick on people, a Syngenta, DuPont, and a Monsanto. And a Dow AgroScience. Did I miss something?
There's some of these patents, like PVAT. No one is stopped from using PVAT for anything because you can get a license from the company and they'll sell you a reasonably priced license when you want it. But then there are other things like, I've heard zinc fingers are this way where the company involved won't give you a license. So, is this technology associated with plants in the category of they're happy to sell you a license when you're ready to commercialize your product, or they use it to prevent you from getting into the market?
They use it to prevent you from getting into the market.
Well, no, I would say it's yes and no. It depends on how they want to see the products coming and whether or not you're talking to the biggest dog on the street or the medium dog or the little dog.
Yeah, so instructive story, Golden Rice, most people are probably familiar with, had, I think, 72 patents associated with it from 40 different companies. And yet they were able to ultimately negotiate royalty-free licenses for each of those, although it wasn't clear that that was going to happen, and probably wouldn't happen with every instance of this in the future. Okay. All right. Other tools needed or under development? June.
So I think some of these parts really need to be standardized as bacterial communities have been.
Yeah, okay, meaning...
Well, is that what you're doing? You're standardizing?
So
there will be a standard 35S promoter?
Well, we use the same standards as the MIT Firebricks, but it may be the Firebrick or anything else, sort of settle on that yet. But they'll essentially use the same, so they're compatible with vectors, etc. We hope to use the same kind of database conventions that Firebricks would use and donate them to the MIT registry or whoever's running the registry. And the only real difference would be that you would have distribution through the plant sources, because there's not much point in stacking the Firebricks repository with stuff which is not particularly relevant for microbial people. So.
Right.
So I'm not a plant biologist, so I was just wondering if you could clear up for me. Is it clear what types of promoters are already established for use in synthetic biology in plants, and are there established reporter genes? Like, is there a plant GFP that people use to measure promoter measurements? This might be a really naive question, but like, what is the current status? Is there a list of promoters that a lot of people use when they put things into plants?
Yeah, there's a number of promoters. Actually, there's a rather restricted set of promoters that people generally use, partly because of access. And outside the academic community and industry, there's a very much wider range of promoters. And companies like Ceres, for example, have libraries of hundreds of tissue-specific and species-specific promoters they have access to. Reporter genes are quite limited, but there are essentially plant-specific. They're fairly broad and general, but you've got the original was the GUS gene, which is a beta-glucuronidase because it's background beta-glycosidase activity in plants.
And the GFPs in dicot species, for example, you can't use wild type GFPs because there's a cryptic intron which gets spliced. But essentially, most elements with high GC content in terms of codon usage work pretty well. So, but you would use something which has been proven and want to use that. And again, these are the kind of things that various labs, including our own, have sort of worked on and have reliable GFPs that work. So
yeah, I think
capitalizing on that.
I think plants have almost the same diversity of reporters. I don't know if it's just my perception, but as far as promoters and terminators go, I think a lot of people use the same promoter, but it's not necessarily the best promoter, right? It's
essentially history and what's available.
It's legacies and histories that everyone uses kind of the same constituent promoter that has been used previously. And so not a lot of work has gone into optimization in some cases of some of these widely used parts.
And if you characterize something in, for example, Arabidopsis, can you translate lessons from Arabidopsis to other plants, or is it variable?
For the most part, yeah, that's...
Well, yeah, it's somewhat. It'll work for a dicot, not for a monocot.
Is there a good way to engage some of these companies with the academic community? Because I think, in some ways, with plants, the commercial entities are much more advanced in terms of their libraries of promoters and genes that they know have specific function, but a lot of that information just isn't currently available. So, whereas on the other side, when we look at the pharmaceutical industry and some of the biotech industry, I don't think that they have as much information that we might like to get our hands on. Is there a way that you can envision to gain access to that information, to partner in a productive way?
Well, a number of companies have already donated considerable resources. For example, Syngenta have a collection of knockouts throughout the Arabidopsis genome, which are a really useful community resource. Series have donated essentially collection of thousands of full-length cDNAs to the community. But because the plant biotech community is quite protective of its IP and uses it as a tool to sort of an anti-competitive element, they are a little reluctant to just broadly distribute things. I think it has to be said. I think it's an understatement. I think.
Yeah, I'm not optimistic, but there may be a possibility if they saw PR value or some pressure from the broader community to throw at least some crumbs the way of the public, right? And they have thousands of promoters characterized, for example, and there may be some that they're on their D lists that they have no plans ever to use. That is the most I can imagine coming. Having said that, though, I know someone who works at a small plant biotech company, and he says that his company, when they want a promoter, they just look for what's expiring in the patent databases that year because they've been well characterized. There was a lot more excitement over patenting promoters, 20, 25 years ago, and now there's a lot of promoters coming off patent that might be useful to incorporate in some of these things.
That might be something that's good to have for the community, which is a list of expired patents and what resources would be open access, essentially.
Does anyone know what's happening at Cambia?
Richard is traveling the world and has hired lawyers rather than scientists.
Right. And that's been going on, though, for... So Cambia, this organization I mentioned at the beginning, did put as much effort, and even now it sounds like more effort into the legal side of things than the scientific side of things, and they've written lots of white papers about what needs to be done to find a way through certain patent landscapes and so on, or the status of certain patent landscapes. And I wondered if they had that type of information already, or if it could be made available to people in synthetic biology who might not know how to find it otherwise. So we can look into what Cambia.
Jim, it's pretty easy to make normal biobricks. How easy is it to make phytobricks if there's a part that you want to use or a gene that's not in the collection?
I guess our plan is to essentially mine the literature because there's a lot of material out there that's been described and characterized. But for instance, in the early days when people were focusing on essentially bashing out and defining functional domains, promoters and the like, which has been done and then buried essentially. That every lab would patent it, locked up by the host university or institution, and they try and negotiate some kind of arrangement with the company, and it's, I think, just to break out of that cycle.
Yeah, 3 hours.
Can be extremely useful for the community, I think.
Yeah. Yeah, but there are no particular reasons why plant biobricks would be any more complicated conceptually than bacterial biobricks. How many fighter bricks do you plan to make?
Well, it's a mixture of PCR-based things and synthesized things, depending on restriction sites and how we do it. So we have, what have we got? $250,000 to make a collection of gram-positive and plant biobricks. So it depends on how we spend it.
Great. Okay. I'll also mention our iGEM team this year. A couple of the members were interested in plants and took one of those Cambia vectors and put the compatible ends on with the Freiburg standard for insertion of promoters, selectable markers, and coding regions. And there's a poster here describing that.
And unlike bacteria, plants use introns. Are there different specificity depending on which plant as to what the splice sites are? Do we have to be concerned about things like that, getting rid of those cryptic sites if they exist? And would these things be compatible across all plants, or would it only be an Arabidopsis only sort of collection?
It would be a dicot monocot division. So those are the two big divisions you have to worry about. So you have to have a phytobrick, and ideally a phytobrick monocot and phytobrick dicot. That would be the big one. Because I assume Jen's going to make phytobrick dicots just because they're easier.
I think one of the other things is that if you're getting heterologous sequences, particularly from something like yeast, which is AT-rich, in dicots anyway, the actual intron recognition is partly the boundary sequences, but it's also the content. So they look for AU content within the body of the intron. And so GFP, for example, which comes from the Icoria victoria jellyfish, is AT-rich. And so when you transcribe it, it recognizes an 84 nucleotide piece in there that gets recognized and spliced out because there are cryptic sites at each end, but it's the content. So anything that you've got that you want to be happily expressed, it's very worthwhile checking for AT content. So if it's got a reasonable GC content, you're probably okay. But monocots have a slightly different mechanism for recognition. They're much more conventional, animal-like, if you care, where they look at the boundary sequences and branch point rather than the content. But that's a serious issue with heterologous expression.
All right. Any suggestions of types of biobricks that you'd like to see made available? Beyond the obvious that have been mentioned already, promoters, tissue-specific promoters, constitutive promoters,